Friends, Assalamu alaikum to all of you in botany class. We were discussing photosynthesis in previous lecture, but I have told you there were two pathways, light reaction and dark reaction. But in case of light reaction, I have told you that light reaction takes place in presence of light, in which ATP and NADPH formation takes place. But in case of this dark reaction, products of dark reaction, products of light reaction are involved. So the process which is taking place in dark reaction do not take place in presence of light. So this phase is called a biosynthetic phase. Food formation of dark reaction has two pathways. We know it is one cycle is called C3 cycle, and it is named after the name of scientist Kelvin. It is also called Kelvin cycle. So when glucose formation takes place, this whole process runs in the form of cycle. So why it is called C3 cycle? We know that in this cycle the first formed stable product has 3 carbon molecules. So it is called C3 cycle. Where is this C3 cycle present? We know that C3 cycle is present in mesophyll cells, and mesophyll cells are present in leaves. These mesophyll cells are specialized cells which carry photosynthetic pigment, chlorophyll pigment. Now there are some substances, and one I will write for example here. It is ribulose 1,5-bisphosphate, in which there are 5 carbons, C5. So it is ribulose 1-phosphate. Its function is to combine with atmospheric CO2, atmospheric carbon dioxide, in presence of rubisco. So rubisco enzyme is the enzyme which has the ability to combine with, it is rubisco carboxylase plus oxygenase. It has the ability to combine with carbon and combine with oxygen. So when this ribulose 1,5-bisphosphate in the presence of rubisco enzyme combines with CO2, it means that in the first step it undergoes carboxylation. So this rubisco enzyme, we know that it is responsible for this reaction. When 5 carbon compound combines with another carbon of CO2, it becomes there becomes 6 carbons. So it becomes C6 carbon compound. So it undergoes carboxylation. It means that addition of CO2 takes place here. Now 6 carbon compound is formed with the help of rubisco enzyme or UBP. Then there is ribulose bisphosphate. It means that I have already told you that it has the ability to combine with carbon and also to combine with oxygen. That is, in case of this reaction, it combines with carbon dioxide, and then 6 becomes 6 carbon compound into 3 carbon molecules of 2. This 6 carbon compound first it becomes, and then it splits, it breaks into 2 carbon molecules. Then the 2 carbon molecules which are formed here, it is called as phosphoglyceric acid, or we can say it is PGA, which is formed as the stable product in this pathway. So in so because of this stable product, it is called as C3 cycle. So this PGA is formed, and we know these are 2 molecules. This PGA is multiplied by 2 because these are 2 molecules. These 2 molecules of carbon are present here. So first step is carboxylation, and second step is reduction. Then we make food with the use of consuming ATP. Then ATP is, when we consume ATP, it means that ATP is converted into ADP plus inorganic phosphate. Now in second step, during reduction, NAD positive is formed from NADPH, which is the product of light reaction. That is, ATP converts into ADP plus inorganic phosphate. So NADP converts into NAD positive. Then we know that there are two molecules of C3C3. So we have triose sugar where C3C3 carbons are present. So any one C3 will release one carbon, and one carbon will be released in each cycle. When we run the cycle, this carbon will be released in each cycle, so that later on one carbon will be reduced in sugar for the formation of that sugar which we want to form. Then when we take this ribulose 1,5-bisphosphate, it means that ribulose 1,5-bisphosphate in presence of Rubisco enzyme combines with CO2. It means that it undergoes carboxylation. Then 6 carbon compound is formed, and this 6 carbon compound breaks, or we can say it splits into two molecules, C3C3. Then there is formed phosphoglyceric acid, PGA, and it was the first step. When this is multiplied by 2, it means that 2 ATPs are used here. So when this C3C3 carbon compound releases one carbon, it remains C5. It means that there are 5 carbons now. Now regeneration takes place. So in this regeneration, when regeneration takes place, regeneration takes place, one ATP is consumed here, and it converts into ADP plus inorganic phosphate. So then a 5 carbon molecule will be formed, and it will be a ribulose 1,5-bisphosphate. So who is ribulose 1,5-bisphosphate? It is the first who have accepted CO2 at first. So we will see here how many ATPs are used here. At first we see there were 2 molecules, and there we have used, it means that we have used 2 ATPs here. And in case of when regeneration takes place, then another ATP is used. It means that when we run a cycle at one time, there is use of 3 ATPs, and when this cycle runs 6 times, it means that 6 is multiplied by 3. So we have got that we have used total 18 ATPs in this Calvin cycle. So once again I will give you a reminder in this, that first of all we will take ribulose 1,5-bisphosphate. Then in presence of Rubisco enzyme, this Rubisco enzyme has the ability to combine with carbon and combine with oxygen. Then at first it combines with carbon dioxide, and it means that it undergoes carboxylation, where C6 compound is formed, the compound which has 6 carbons. Then this 6 carbon compound breaks into 2 molecules. It means that C3C3, which is called as phosphoglyceric acid, and in short we can say it is PGA. This PGA undergoes reduction. In this reduction, with the use of ATP, it converts into ADP plus inorganic phosphate, and NADPH converts into NAD positive. Then there is triose sugar. There are 2 molecules of C3C3. So one C3 will release one carbon So carbon is released by one molecule, then it goes under and third step is it goes under regeneration, in which with the use of ATP, this ADP plus inorganic phosphate is formed. So we have got ribulose 1,5 bisphosphate once again, which is the which has who has accepted this CO2 at first. So when we run this cycle, at one time we have taken 3 ATPs, then when we run this cycle 6 times, and it means that 6 is multiplied by 3, there is total use of 18 ATPs. So it was all about this photosynthesis. Now next time we have to take another pathway of this photosynthesis. Thank you very much.